I'd like to introduce the next speaker. It's Dr. Anthony Ratcliffe. He's the president and CEO of Synthasome Incorporated. Dr. Ratcliffe.
Thank you. So I'm going to take a leap from the basic science that's trying to move into translation and talk to you about translation and product. And I think you'll all be able to work with me and understand where perhaps the rubber really meets the road. So I guess we'll go that way. Okay, so here's some opportunities. I'm going to talk about skin and musculoskeletal diseases. And so you can see that we have some opportunities. Here's skin, some chronic wounds, burns, and EB. And remember little Dominic here, just one slide later. Articular cartilage for osteoarthritis, trauma, sports, tendon and ligament, sports medicine, foot and ankle, hand. You've all seen rotator cuff, ACL, Achilles, the Tommy John procedure, all of these types of issues. There's neuromuscular diseases, and we'll hear more about that in the speaker after this. And there's bone, craniofacial, as well as long bones, trauma, tumors, and atrophy. So we've got lots of opportunities where you could see that a cell-based therapy could have an effect. So here's some products that are already on the market. So Carticel, generated by Genzyme for articular cartilage repair, came out in 1997. Transcyte, developed by ATS, where I was, brought out a burn product in 1997, and Apligraf from Organogenesis brought out a viable skin product for chronic wounds in 1999, and ATS brought out Dermagraft in 2001, again for chronic wounds. Orcell came along also with another product for chronic wounds. So, what about profit? Okay, that's a problem. Now, you understand, right? Dermagraft actually generated profit in the end because it went through a couple of cycles of being bought, and then Advanced Biohealing sold it to Shire, and they made money. They were several cycles down. The people who invested and put in their time and effort right at the beginning certainly didn't make anything. And Apligraf by Organogenesis came out of bankruptcy, and I think they're beginning to make some money there now. So these two cell-based products used for chronic wounds have managed to creep into the profit. But if you're going to have a product on the market, it has to make money. And this is a new feature. Beyond does it work is how do we make it commercially viable? Here's the regulatory pathways that we can use in the US. And we've got timelines and we've got dollar amounts. For a 510K pathway, it's going to cost you between $5 and $20 million in 2 to 3 years. The time point is when you're starting your key animal study, for example. If it's going to be a device, it could cost you $30 to $200 million and about 6 years. Biologics and drugs, it's going to cost you 50 to more than 300 million, you choose your number, and it's going to take you 8, 9, 10, 12 years, whichever you want to choose. I'm sure everybody can provide me with an example where they can do it for less money and or a shorter period of time, and they may well be correct. These are just general, but for each one of those I can provide you with 10 that says it takes longer and costs more money. So this is where we live. This is what we've got to be able to do to generate a product. Allografts are already in this mix. Allografts, if you want an allograft product, it can cost you less than $50,000 to have that as a product line in your company. In fact, earlier this week, I went to advise a company who was just doing exactly this. And they were bringing in skin substitutes and amniotic membrane for wound healing. This is low-hanging fruit. They'll pick off some of the market. It may work, may not work, but it really provides a challenge for those products that are going to be able to address all of those wounds and generate a successful repair. Same things applying in bone, articular cartilage, tendons, and ligaments. Of course, these are really important features. Skin as an allograft is also an important feature, but we've got to put that in the mix and understand where we are with that, because it certainly is a competing product for cell-based therapy. Now, I talked about profit. Here's the real point. You've got all that money you've got to spend. You've got to get a return on your investment, right? So this is your development costs, and we've just seen how much that might cost. But you've got to balance that with what your income is and what's your cost to manufacture and sell, and then you build in some other features like risk. And we've got to avoid this, which is where the R&D, which is our cost, outweighs the revenue. And this is your challenge. Bear it in mind. Okay, quick examples. That was a bit deflating, wasn't it? How much money are we going to have to raise? You know, the standard model is you do your R&D, and NIH is really great at generating the start of all of that, and they say, okay, now you go to venture capital. Venture capital is a misnomer nowadays. It's the wrong word because they don't venture. So how are we going to do this? Well, I want to bring some history here. This is Dermagraft. Remember, this came out in 2001. It's cells, dermal fibroblasts seated on a scaffold, grown in a bioreactor. We then cryopreserve this. It's thawed in the doctor's office or in the clinic, and it's applied to a chronic wound. This is how it works. You put it on a series of weeks, and then you get a great result. It's an excellent product. Apligraf does a similar kind of thing. So we can do cell-based therapy. Where are some of the challenges, and where are some of the opportunities? This is the list that you would like to see if you're taking a photograph or taking notes. This is the one that you probably want. I want you to remember that cost one, you know, the 5 million, 30 million, more than 300 million. But here's the opportunities. So one, we've got to figure out how to drive down our cost. To do that, we've got to increase efficiencies. That means talking to organizations such as the FDA. They do a phenomenal job, I believe. I'm not an FDA basher. There are people. I am a great supporter of the FDA. Incredible job under you know, challenging rules and regulations. So we need to be able to work with the FDA and other organizations to really increase our efficiency. Part of that is standards, and you'll see that standards are mentioned throughout here. Just to mention, I am a co-chair of a standards committee at ASTM on tissue-engineered medical products, so of course I'm biased, but I'm that co-chair because I think it's important. Okay, we've got to drive down cost. We've got to shorten the time. Time is money as well. And as a CEO, I can tell you that if we've got to go away and find more money, that doesn't work, or more time, it's really difficult. So we need to streamline our studies, really try and make that time as short as possible. Technical difficulty, we've talked about that already. Cell source is the issue. Standards can help immensely with that. Clinical uncertainty. Of course, we've got to do those clinical trials. But why do we see clinical trials struggle so much? Well, part of the deal is that we don't really know what the data is out there. I'd love to see access to improved clinical databases. We're all generating clinical databases. As a patient, I'm looking at this age group in here, you're all patients. So have you noticed you're going to the doctor more and more? So you're part of a clinical database somewhere, but we don't have access to that. As an industry, we want to have access so we can accurately target what we can deal with and what we don't have to deal with. There are regulatory uncertainties. We already mentioned that, but standards can really help make that smooth. Manufacturing. Scalability is a big issue, and I'll touch on that in a minute, and standards will help with that too. We've got to look at our market opportunity. Lots of people stand at the front and they say, well, I'm going to do $1 billion. It's not $1 billion. It's a real small number usually, and we need to be accurate about that. We need availability of data. It goes back to the databases. How many patients are there really? How many can we treat? And then finally, can we use international opportunities? Well, I can tell you that here in the United States, we do the FDA thing, we get our approval, but then we're going to go to Europe and we've got to do a different thing altogether. And so we need harmonization of that to really execute. Scalability. I'm just mentioning a few topics just to bring this to play. And we've already heard some of this. This is cell culture. in the research environment. And you can see that you've got a Petri dish, and then you get a flask, and then you get roller bottles. And this is what happens in a research lab. We mentioned academic environments have GMP facilities, but it's not necessarily a scalable system. This is research type stuff. If you're going to address tens, hundreds of thousands of patients per year, you need to be in an efficient manufacturing system. On the top left, that's roller bottles. They're working, right? In the middle, you've got robotics helping us change media, harvest cells. Here at the bottom, we've got bioreactors, and they're manifolded, they're minimizing contact with humans. The first slide, the first image, I just took off Google. The last two slides, they're over 20 years old. This is not new technology. That's what we did at ATS 20 years ago. So we can do scale up. It's there. We need to share and acknowledge that all of that occurs. But you have to do scale up in a proper way, otherwise you won't meet the needs and drive down the cost. There is some advance here. Astrolab is developing a cartridge product. It's autologous, and what they've done is they're taking a cartridge biopsy, they're putting it in a device that isolates the cells from the cartridge, expands those cells, seeds them onto a scaffold, grows them for a little period of time, and then can implant that. Now that's a sealed unit, and scale up means that you just use more of those sealed units. And this is the next advance that I'm seeing in manufacturing and allows autologous use. I mentioned standards. Here's an animal model for articular cartilage repair. This is one that we're doing. It's NIH funded. They saw the clear need, and so NIH put some funds here. And this is an example of a large animal standard that we're developing. It'll go and become an ASTM standard. And then we'll have some uniformity about articular cartilage type approaches. We've heard about autologous and allogeneic cell sources, and they tend to just get lost in the system. These are two very, very different business models. And if we're going down the autologous route, you have to take on that approach somewhere that we've just seen and make it scalable. Allogeneic is scalable, as already shown. You've got robotics, etc. But we've got some other challenges. So bear in mind those two are very different. The area of skin and orthopedics, it's often going to be targeted rather than systemic, so you'll end up with scaffolds, etc. And a lot of this has already been done. What we can do is we can do biomimetics, so that we can use biomaterials to provide some of the features, for example, mechanical properties. And now we want to address bringing cells and matrix to that. And then we've got the ideal issue. But we do have to address some other market issues there so that we're represented on a clear playing field. But this is where we can go as long as we can manage that and make it a good return on investment. Okay, here's the final message. There's a challenge, and that's to develop a treatment that's therapeutically effective, and we hear a lot about that. We also have to meet the challenge of developing a product that's commercially successful. And that's really an equal challenge. Don't underestimate how difficult that is. To have clinical impact, the challenge is to develop a product that is therapeutically effective and commercially successful. And that's the piece I'll leave you with. Thank you.